Good morning, everyone. Today we will discuss about lipoprotein metabolism. So, as you know, these lipoproteins can come under example of conjugated lipids as well as conjugated proteins. It will be a mixture of lipid and protein, and they are helpful in transportation of lipid across various organs. So, in the chapter of digestion and absorption, we have learned that major lipids post digestion includes triacylglycerol, phospholipids, free cholesterol, esterified cholesterol, and free fatty acid. Okay. Majority will be in the form of cholesterol esters, phospholipids, and triacylglycerol. Okay. Now we know that lipids are insoluble in aqueous phase. Okay. So for their transportation, we require help of proteins. And combinedly, these lipid and proteins are known as lipoproteins. So main function of this lipoprotein will be associated with transportation of lipids. Here, the protein part of lipoprotein is known as apolipoproteins or apoproteins. There are free fatty acids. They form complex with the albumin. They are not considered under the heading of lipoproteins because the complex of albumin and free fatty acid is very loose and can be easily detached. Okay, so protein part of lipoprotein will be known as the apoproteins or apolipoproteins, and combinedly along with the lipid they are known as the lipoproteins. So if we see the basic structure of lipoprotein, so in lipoprotein structure it will be kind of mixtures of our digested product. So it will be a mixture of say free cholesterol, there will be cholesterol esters. If you see in the center part, there are cholesterol esters. Along with that, in the peripheral part, there will be phospholipids, and in the center core, another main important thing will be the triacylglycerol. Okay, so the basic structure of lipoprotein you should know and try to draw the simple diagram of lipoprotein in case of if there is a long question on lipoprotein. Okay, so in the center you have to show the triacylglycerol as well as the cholesterol esters. Okay, the free cholesterol is present in the bilayer. Okay, so that is the difference. Cholesterol esters in the center and free cholesterol is in the bilayer. So bilayer will be made up of phospholipid. Now coming to the lipid protein part, there are two varieties of protein present. One is the peripheral protein, another one is the integral protein. So peripheral protein, they will be loosely attached with the lipid part, and they can be easily exchanged between various lipoproteins. In contrast, the integral apoprotein such as the ApoB48 or ApoB100, similarly ApoA, they form integral part of structure of lipoproteins. Okay. So they are very tightly bound and they form a very integral part of a structure of a lipoprotein. In contrast, the peripheral apoproteins, they are mainly responsible for the activation of certain lipoprotein, and they are easily exchanged between lipoproteins such as the HDL and LDL. So this is the same picture which is showing the structure of lipoprotein. Another thing which I already told you to remember is that unesterified cholesterol or free cholesterol is present in the bilayer, which is made up of phospholipids. Okay, cholesterol esters and triacylglycerol, they form integral of lipoprotein. Also, we will have a protein at the periphery as well as in the integral part. Okay. So you have to remember the basic structure of lipoprotein. Coming to the classes of lipoprotein or classification, so mainly they are divided into five categories: chylomicrons, VLDL or very low density lipoprotein, LDL low density lipoprotein, HDL high density lipoprotein, IDL intermediate density lipoprotein. So here if you see, the common word in most of the lipoprotein is the density. So their classification is actually based on the density, and that density is decided by the ratio of lipids as well as the protein in the structure of lipoprotein. So as we know that lipids are less dense than protein, so the lipoprotein which will have more lipid content will have low density. In contrast, the lipoprotein which will have more protein content, it will have more density. Okay. So more the protein content, more dense the lipoprotein. So now according to the name, we can easily say that the HDL, that is the high density lipoprotein, will have more of protein in comparison to lipid. Okay. In contrast, the LDL or VLDL, they will have more lipid in compared to carb, sorry, protein. So their density will be low density and very low density lipoprotein in compared to other lipoproteins. Okay, so chylomicron is the largest lipoprotein in the size, but its density is lowest. That means chylomicron will have a very high lipid minimal of protein. In contrast, HDL will be smallest in the size but will have highest density. So it will have a more protein. So if you see the composition or the ratios of lipid and protein in all the lipoproteins, we can see here that in chylomicron, the lipid percentage is almost 98 to 99, and remaining only 1 or 2% will be the protein. In contrast, in HDL, if we see, the protein content is maximum almost 40 to 50% of composition of HDL is made up of protein. In contrast, the lipid portion is around 40 to 60 degree. So as we move on from chylomicron to HDL side, the lipid content is gradually decreasing and protein content is gradually increasing. Okay, fine. So this is the diagrammatic, or you can say a graphical representation of the same thing. The yellow part represents the triacylglycerol, the orange part represents the cholesterol. Okay, this light blue part represents the phospholipid content, of any lipoprotein, and dark blue part represents the protein, okay, which is already shown in this particular slide. Okay, so based on that, chylomicron is the largest in the size, but the density of the chylomicron is lowest. In contrast, HDL is the smallest in size, its density is the highest. Similarly, the separation of lipoprotein can be done by electrophoresis. So electrophoresis is the process where it separates the various proteins based on the charge. So in lipoprotein, there will be variation in the protein percentage. So the charge of various lipoprotein also will be different, isn't it? Okay. So that's why the chylomicron will have a lowest of protein, so it will not move much. Okay, it will move very slowly. In contrast, HDL with maximum protein, it will move furthest among all the lipoproteins. So this is the composition of lipoprotein. You have to remember approximate number of the content of TG, cholesterol, as well as the protein part. So if you apply logically, then also you can remember the table that in case of chylomicron, you have to show the maximum percentage of triacylglycerol, lowest percentage of the proteins. Okay. Similarly, in case of LDL, you have to show the maximum content of the cholesterol. In HDL, the maximum content will be of protein. Okay. So even if you don't remember the exact number, the ratio of triacylglycerol should be maximum in chylomicron, ratio of cholesterol should be maximum in case of LDL, and ratio of protein should be maximum in case of HDL. That basic thing you have to remember. Okay, so this is the same way a pie chart has been shown, which is showing the composition of various lipoproteins. Now coming to the protein part, which is known as the apoprotein or apolipoprotein. There are many varieties, more than 10 types of protein have been identified in the structure of lipoprotein, but we have to remember mainly 3 to 4 varieties of apolipoprotein, as we know that they are must for transportation of lipids in the circulation. There are two varieties of protein present in structure of lipoprotein. One, the integral lipoproteins. Examples are ApoB100, ApoB48, ApoA1. Peripheral apoproteins, which includes ApoC and ApoE. Okay. So these are the various apoproteins or apolipoproteins. More than 10 varieties have been identified. Each one have their own significance, but for the UG purpose, you need to remember these three: ApoA1, ApoB100, and ApoB48. So remember here, ApoA is associated with the HDL, and it is necessary for activation of one particular enzyme, which is known as the LCAT. Okay, the function of LCAT we will discuss separately in HDL metabolism. ApoB100 is associated with the LDL activity, and ApoB48 is must for the chylomicron formation. Now, even in recent NEET PG exams, there was a question asked regarding ApoB100 is associated with which lipoprotein, and then there were these four options were there. Okay, so the correct answer was LDL. Okay, ApoB100 is associated with the LDL. Okay, now coming on to the individual metabolism. Okay, so first we will discuss about metabolism of chylomicrons. So the chylomicrons, which are synthesized in intestinal mucosal cell. So the site for the synthesis of chylomicron is intestine mucosal cell. As we know that it will have a 98 to 99% of lipid in its structure, majority of that lipid will be in the form of triacyl glycerol, and the protein content is around 1 to 2%, and the major protein which is present in chylomicron is ApoB48. Okay, ApoB48. The function is must for you to remember of any lipoprotein, and based on the function you can even recall the metabolism of a particular lipoprotein. So function of chylomicron is that they transport exogenous, that means the dietary lipids, the lipids that we consume in the form of diet. They will transport that lipids for the utilization to the peripheral tissues, from intestine to peripheral tissues such as the heart, muscle, adipose tissue, kidney, etc. etc. Okay, so chylomicron represents the exogenous fat which we consume in the form of diet. Okay, and it will transport that fat to the various peripheral tissues. Now, coming on to the metabolism of chylomicron. So, this particular slide you can refer once we discuss this particular image. Okay. So, the site of the synthesis of lipid or chylomicron will be the smooth endoplasmic reticulum. Okay. Smooth endoplasmic reticulum synthesizes the lipid. It will receive the protein part from the RER, that is the rough endoplasmic reticulum. Okay, so the lipids has been synthesized in smooth endoplasmic reticulum. Protein part will be received from the RER, that is the rough endoplasmic reticulum. They will combine, lipid and protein will combine. They will move towards the Golgi apparatus, where minor carbohydrate part also will be attached to it. Okay, so SER plus RER and Golgi apparatus combinedly will form lipoprotein. The site will be the intestinal lumen cells. After that, these lipoproteins will be accumulated into the lymphatic vessel. Ultimately, they will be drained into the thoracic duct, and from thoracic duct, they will come into the circulation, and from that they will be transferred to the peripheral tissues. Okay, so this particular thing we have already discussed in digestion and absorption, that digested lipids, they will be packed in the form of chylomicron. Okay, digested lipids, okay, packing will be done in the form of chylomicron, and chylomicron will come into the thoracic duct, and then from thoracic duct it goes into the various capillaries or into the circulation. Okay, so this is the pathway of circulation of chylomicron. Okay, so this particular slide has been described in detail in here, that the site of the synthesis of chylomicron will be the intestine. Okay, then there will be role of SER or RER. From RER, it gets its protein part, that is the ApoB48. From Golgi apparatus, it receives its carbohydrate attachment. Then the chylomicron, okay, whose structure mainly will be consist of ApoB48, that is the protein part. Lipid part will be triacylglycerol, and then there will be minor carbohydrate. It will enter into the lymphatic circulation, that means in the thoracic duct, and then it comes into the blood circulation. Okay, so this type of chylomicron is known as the nascent chylomicron. Okay, remember the word nascent. So the nascent chylomicron will have ApoB48 triacyl glycerol, mainly in the form of lipid composition. Along with that, it will have cholesterol esters and some amount of free cholesterol. Okay, so this is the first part in the synthesis of chylomicron or chylomicron metabolism. After that, this nascent chylomicron will receive few other apoproteins, that is the ApoC2 and ApoE, that it will receive from another lipoprotein HDL. Okay, so as we discussed that ApoE and ApoC, they are the peripheral proteins present in the lipoprotein. So they will be easily exchanged among the various lipoprotein. So chylomicron, that is nascent chylomicron, will receive ApoC2 and ApoE, that is from HDL, and it becomes the mature chylomicron. Okay, so mature chylomicron will have ApoC2 and ApoE, which is received from HDL, along with the ApoB48 and mainly Tg in the internal core. Okay. Now this mature chylomicron will come into the capillaries of various peripheral tissues. Suppose if we take example of adipose tissue. Okay. So adipose tissue will have a hormone which is known as the lipoprotein lipase. So as its name suggests, what does it will do? It will hydrolyze the content of lipoprotein. And here our lipoprotein is chylomicron. Its content is PAG, cholesterol esters, as well as the cholesterol. Okay, so lipoprotein lipase will hydrolyze this triacylglycerol mainly into glycerol and free fatty acid. Okay, so in adipose tissue there will be lipoprotein lipase. It will be activated by the presence of ApoC2. Okay, ApoC2 will make it activate, will do the activation of lipoprotein lipase. So lipoprotein lipase will deliver the free fatty acid to the peripheral tissues, for example to the adipose tissue. Glycerol part will come into the liver. Okay, adipose tissue cannot utilize glycerol, reason you already know. Okay, fine. So after delivering the free fatty acid and glycerol to the various peripheral tissues, the Tg content will be reduced from the mature chylomicron. Also, there will be removal of ApoC2 and it will be delivered back to HDL. Okay, so ApoC2 and ApoE, which was received from HDL to the chylomicron, now chylomicron will revert back the ApoC2 part, but ApoE part will remain with the chylomicron. And this type of chylomicron, which will have a low Tg, ApoE and ApoB48, is known as the chylomicron remnant. Okay, so the purpose of the chylomicron, which was to deliver the dietary lipids to the peripheral tissue, has been achieved, and remaining part is known as the chylomicron remnant. It will goes into the liver, and liver will have a specific another enzyme which is known as the hepatic lipase. Okay, that will hydrolyze the cholesterol esters, and ApoB48. Okay, so the cholesterol esters will be hydrolyzed, so there will be cholesterol formation. And proteins, ApoB48 are proteins, so once they are hydrolyzed, they will produce amino acids. Okay, so in this form, the dietary cholesterol which we have taken is delivered to the liver. Okay, so that's why this is the role of chylomicron, that it has delivered all the dietary lipids such as the triacylglycerol or cholesterol esters, cholesterol to the peripheral tissues and finally to the liver. Okay, fine. So this is the main function of chylomicron that you have to remember. And if you apply basic sense and logic associated with the function, then you can easily remember the metabolism of chylomicron. Okay, so this is another way to represent the same cycle of chylomicron. Okay, you can also explain the chylomicron cycle by this particular type of flow chart. Now you will come across the various lipases. You already know about the lipoprotein lipase. Its action or what does it do is already explained in this particular slide. So lipoprotein lipase, which will be present in the capillary walls, it will act on the triacylglycerol of chylomicron and VLDL. We will have another variety, hormone sensitive lipase. It will be present in the adipose tissue. Where does it act? It acts on the triacylglycerol which are stored in the adipose tissue. Okay, insulin will activate lipoprotein lipase and insulin will inhibit the hormone sensitive lipase. Okay, remember the difference here. Lipoprotein lipase will hydrolyze the triacylglycerol of lipoproteins. Hormone sensitive lipase will act on the stored TG. Okay, hepatic lipase in another variety which is important in lipoprotein metabolism. The site will be the liver. Where does it act? It acts on the triacylglycerol of VLDL remnant, chylomicron remnant, on IDL, on HDL. Okay, so the all the three lipases will hydrolyze the triacylglycerol, but their site of action is different. Okay, so please look the carefully and try to remember this slide. It will helpful in understanding the integration of metabolism as well as the lipoprotein metabolism. The second important lipoprotein is the VLDL. Now VLDL and chylomicron both undergoes same type of metabolism. The minor difference is something like this, that the VLDL will be synthesized in the liver. Major lipid here also will be the triacylglycerol. The protein which is necessary here is ApoB100. In chylomicrons, it was ApoB48. The major difference here will be in the function, that VLDL is associated with the transportation of endogenous lipid from liver to peripheral tissues. In chylomicron, it was dietary lipids, that is exogenous. Here, VLDL transports the TG which is synthesized by liver, and it will deliver this triacylglycerol to various peripheral tissues. Okay, so this is the main difference. Production and processing is more or less same like chylomicron, so we are not going into much detail. Only thing will be here is that the protein part will be the different, and the site of synthesis will be liver in contrast to intestine. Okay, so all other things is similar up to nascent, sorry, up to VLDL remnant part. Okay, so just like chylomicron, it will receive the ApoC2 and E from HDL. Then there will be action of lipoprotein lipase. Okay, there will be release of free fatty acid, glycerol, and remaining part of VLDL with less TG is known as the VLDL remnant. Another name of VLDL remnant is IDL, that is the intermediary density lipoprotein. From IDL, the ApoC2 will go back towards HDL along with the ApoE. So remaining ApoB100. Okay, after that there will be addition of ApoB100. So ApoB100 and the VLDL with low TG, that is known as the IDL, will have a two fat. One, it forms the LDL, whose metabolism we will discuss in next class in detail, and remaining IDL will go towards liver, and by the action of hepatic lipase, it will be converted into cholesterol and amino acid and free fatty acids. Okay, so VLDL metabolism is very similar to chylomicron metabolism. Okay, and that thing you don't have to remember in detail about VLDL metabolism. Only thing which you have to focus in VLDL is this particular slide, that is the site of synthesis of VLDL, major protein present in VLDL, and its function, how it differs from the chylomicron. Okay, fine. So rest of the thing we will discuss in our next class. Okay, about metabolism of LDL as well as the HDL. Okay, so thank you very much.